Hello, and welcome to the second segment of Cardiovascular Physiology Part 3. Where we left off was on slide 58. And so for the entirety of this segment, we will be talking about hemostasis and anti-clotting systems. This will run slide 58 all the way to the end of Part 3, and that will be the conclusion of our coverage of the cardiovascular system. So hemostasis refers to all of the mechanisms that are in place to stop bleeding when the wall of a blood vessel has been injured or torn open. And there are three distinct but somewhat overlapping mechanisms: vascular spasm, a platelet plug, and clotting. And these are each unique mechanisms, but they do overlap in time. They can all co-occur, and to varying degrees, they assist each other. So vascular spasm, vascular means blood vessel. Spasm refers to muscle contraction. So during vascular spasm, the smooth muscle in a blood vessel, like an artery or an arteriole or even the wall of a vein, contracts to induce vasoconstriction. And that vasoconstriction will reduce the flow of blood through that vessel. So what causes vascular spasm? There are a couple of different ways of triggering contraction of the wall of a blood vessel. One of the mechanisms is through chemical signals, a group of chemical signals called cytokines. Cytokines are a very diverse group of molecules. They're all signaling molecules. They all have different functions. Many of them, not all of them, many of them play a role in the immune system. So they can influence the behavior of certain white blood cells. They can influence the diameter of blood vessels. And so that's why we're talking about cytokines here. Some cytokines that get secreted by injured tissue cells as well as platelets can cause the smooth muscle cells in the wall of a blood vessel to constrict, leading to vasoconstriction. In this circumstance, it's also called vascular spasm. Another trigger for vascular spasm is an autonomic reflex. So there are pain receptors, that's the source of the sensory input in this autonomic reflex, and the motor output is through sympathetic stimulation to the smooth muscle of the blood vessel wall. It causes vasoconstriction. In this diagram, we can see a vasodilated vessel and then that same vessel following vasoconstriction, or in this case, we're calling it vascular spasm because a painful stimulus has led to vasoconstriction. I like this diagram because it includes the innervation, the sympathetic innervation to blood vessels. The second mechanism of hemostasis is the formation of a platelet plug. So platelets are one of the three basic types of formed elements, along with red blood cells and white blood cells. And platelets are obviously involved in the formation of a platelet plug. They also contribute to vascular spasm as well as clotting, the third mechanism of hemostasis. So platelets form in red bone marrow, and they are frequently described as cell fragments. So they form from a larger cell in red bone marrow known as a megakaryocyte that breaks up into cell fragments, and each cell fragment is a platelet. And during the formation of a platelet plug, these platelets are going to aggregate. They're going to clump together to block the outflow of blood through the damaged vessel wall. There are three steps involved in the formation of a platelet plug. Step 1 is platelet adhesion. Step 2 is the platelet release reaction. And step 3 is platelet aggregation. In this diagram, we can see all three steps. In the first panel, which is in the top left portion of the field, we see that platelets are sticking to exposed collagen fibers and damaged endothelium. So if you'll recall, endothelium lines your entire cardiovascular system. It's a single layer of simple squamous epithelial tissue. And so normally blood only comes into contact with intact endothelium. Should that endothelium become disrupted or damaged, it exposes underlying molecules like collagen, which is part of connective tissue. When platelets are exposed to something other than intact endothelium, they'll stick to it, and that's what platelet adhesion is. Moving down to panel number 2, bottom left, we see the platelet release reaction. And the platelet release reaction involves 2 components. Number 1, the platelets undergo a radical transformation in shape. They start off being sort of amorphous, but they have all smooth, rounded edges. And during the platelet release reaction, they become very jagged and spiky in appearance. They develop these long radiating processes. And those processes help them to stick to each other, to get caught up on the wall of a blood vessel. And then the second component of the platelet release reaction is exocytosis. So platelets are full of a variety of vesicles, these spherical sacs that store signaling molecules. And during the platelet release reaction, a variety of different signaling molecules are released into the local environment. And some of those signaling molecules are important for vascular spasm. Some of those signaling molecules are important for the formation of a clot. But here we're talking about the formation of a platelet plug. So some of those signaling molecules can recruit additional platelets to undergo the platelet release reaction. And moving into the third panel, we see our very spiky platelets are getting caught up on each other, and they are forming a cluster or a plug to block the outflow of blood through that damaged vessel wall. All right, so just to reinforce this, during the platelet release reaction, A, platelets extend many projections. They take on a very jagged, spiky appearance. And B, they release many different types of signaling molecules through exocytosis. Some of these signaling molecules activate neighboring platelets and bring them into the formation of the platelet plug. Some of these signaling molecules can induce vascular spasm. And some of these signaling molecules contribute to the clotting process, and they're called clotting factors. I highly recommend you spend 2 minutes watching this animation of platelet activation and the formation of a platelet plug. Our third mechanism of hemostasis is the formation of a clot, also known as coagulation. And clotting is a very complex mechanism, and it has taken decades to figure out the process by which a clot forms. And there are still some lingering questions. It's not a process that we, the body of science, understand completely, although we do understand it fairly well. So the superficial overview here is that during clotting, a fibrin mesh forms in the blood. Fibrin is a fibrous, stringy protein, and that fibrous, stringy protein is going to form a clot. It's like a net that can catch formed elements and prevent them from exiting the damaged blood vessel wall. So it will catch red blood cells, it will catch platelets, it will catch white blood cells, and it forms this hard mass that blocks the outflow of blood. So it's a complicated process, as I mentioned, and there are dozens of substances that are required to form this fibrin mesh, and collectively they're all known as clotting factors. Here we're looking at the end product. So this is a scanning electron micrograph. It's artificially colorized. Red blood cells are actually naturally red, but fibrin I don't think is actually naturally green. But that's what we see here. We see these stringy fibrin threads, and we can see that those formed elements are trapped. They can't go anywhere. There are dozens of different clotting factors, and it has taken decades to figure out the mechanisms of blood clotting. Many of these clotting factors are named by Roman numerals, so a little bit later we might talk about clotting factor 10 or clotting factor 8. And it is important to know that these clotting factors were named in order of discovery, and they do not reflect order of participation in the cascade of chemical reactions that gives rise to a clot. Other clotting factors include calcium ions, which are present in blood plasma, and then signaling molecules that are secreted during the platelet release reaction, and signaling molecules that are secreted by damaged tissue cells. So the clotting cascades are quite complicated, and perhaps you'll run across this again in the future downstream in your education. So I'll give you a superficial overview. Even today it'll sound pretty complex, but believe me, it's not as complex as it is actually in reality. So clotting comes about through a cascade of reactions, and we've encountered that word before, cascade. We encountered it when we were talking about cell signaling, specifically signal transduction pathways, and maybe you remember the term phosphorylation cascade. During a phosphorylation cascade, that happens inside the cell, where one protein phosphorylates another, that next protein phosphorylates the next, and so on. That's a cascade of reactions. And a cascade occurs during clotting as well, except this cascade occurs extracellularly. It occurs in blood plasma. As one clotting factor becomes activated, and it activates the next clotting factor, and so on down the line. And our ultimate goal is to induce the precipitation of fibrin. So these clotting factors, many of them exist in our blood all the time in their inactive form. They're produced by the liver, and they're always in our blood plasma. And they are only activated during the formation of a clot. So we'll encounter these two word endings, or word beginning. One is a word beginning, the prefix pro, and the suffix, that's a word ending, the suffix ogen. And in all of biology, whenever you see the prefix pro, a word begins with pro, or a word ends in ogen, it's a good idea to wonder if those are inactive precursor molecules. They need to be activated in order to have any effect in whatever system you're thinking about. So blood clotting in all of its glorious complexity is initiated by one of two pathways, the intrinsic pathway or the extrinsic pathway. And under most scenarios, they're initiated at the same time. But it doesn't matter. Either one will lead to the formation of this fibrin mesh. And so here's our flow chart of sorts. We'll go through the super basic overview of the extrinsic pathway, and then we'll go through the overview of the intrinsic pathway. And you can see that both pathways lead to the formation of prothrombinase. There's that prefix pro. Pro suggests that this is an inactive precursor. Ase suggests it's an enzyme. So prothrombinase is an inactive precursor to an enzyme called thrombin. And once we achieve the formation of prothrombinase, then all of the clotting cascade reactions that occur from there are part of what's known as the common pathway. And the common pathway leads to the formation of the fibrin mesh. So we'll go through an overview of the extrinsic and the intrinsic clotting pathways. The basic difference, there are two basic differences. One is, what's the source of the stimulus? What triggers each? It's a pretty different stimulus in each case. And then number two, how long do they take? The extrinsic pathway has fewer steps, and it occurs much more quickly than the intrinsic pathway. All right, so the extrinsic pathway occurs when tissue factor leaks into the blood. Tissue factor is a signaling molecule with a very stupid name, in my humble opinion. Whoever named it tissue factor should be demoted. And I just say that sort of half jokingly because the term tissue factor provides no information about what this molecule does. And I'll tell you what this molecule does. This molecule is secreted by damaged tissue cells, so connective tissue cells, muscle tissue cells, fat tissue cells, following an injury to that tissue. And tissue factor can leak into the blood. This is why it's called the extrinsic pathway, because something extrinsic to the blood and the blood vessel wall, something from the outside, has to get into the blood in order to initiate this pathway. And so the extrinsic pathway might occur, you can imagine if you fall down and you scrape up your knee, think about the tissues that are injured there. You're bleeding, so the blood vessel walls are damaged, they've been torn open. Your epidermis is damaged, your dermis is damaged. The dermis of your skin is where we encounter our first blood vessels. And your dermis is made up of several different tissues, mostly dense irregular connective tissue, but there are blood vessels, there are nerves, there are scattered adipose cells, and all of those cells are able to secrete tissue factor. And so that tissue factor leaks into the blood, and it's a pretty reliable indicator that injury has actually occurred and we need to form a clot very quickly. And so this tissue factor, once it gets inside your blood, it interacts with calcium ions to lead to what's known as activated clotting factor 10. And clotting factor 10, once it's activated, will cause the formation of prothrombinase. And once we get to prothrombinase, we're at the common pathway, which we'll talk about later. So in this diagram, I've covered up the intrinsic pathway for now. That will allow us to focus on the extrinsic pathway. So in the diagram, we can see at the top left that that blood vessel wall is torn open. It's damaged all the way through, and that allows an opportunity for tissue factor to leak into the blood. And once tissue factor gets into the blood, it interacts with calcium ions, and that all leads to the formation of prothrombinase. From prothrombinase onward, in this case downward in this diagram, that will be the common pathway, and the common pathway will lead to the formation of fibrin. The intrinsic pathway involves more steps, and it happens more slowly. The initial stimulus for the beginning of the intrinsic pathway is damage to the interior wall of a blood vessel. So you recall that your entire cardiovascular system is lined with endothelium, and blood normally only, quote, air quotes, sees this smooth, perfectly intact endothelial lining of the chambers of your heart and all your blood vessels. Well, that endothelial lining can be damaged, and that may or may not indicate that a clot is required, but certainly it needs to be repaired. And so the stimulus here, the trigger to initiate the intrinsic pathway, is exposed collagen fibers that make up part of the blood vessel wall. So imagine that endothelial lining, and then imagine it being torn up or damaged. Just superficial to the endothelium is a basement membrane, and superficial to that is connective tissue. And connective tissue includes collagen. Collagen is a protein. It's one of the most abundant proteins in your body. It's in all of our connective tissues. And when blood is exposed to these collagen fibers, it leads to the formation of activated factor 10, and that pathway is called the intrinsic pathway. The intrinsic pathway similarly leads to the formation of prothrombinase. And from prothrombinase downward in this diagram, we call that the common pathway, which we'll talk about next. But first, going back to this diagram, we can see a blood vessel wall there, and just the interior, the lining of the blood vessel is damaged. Some of you might be asking, well, Laura, how does that occur? So let's say you smack the back of your hand into the wall. You're not bleeding. You're not even bruising. But it's possible that you could have caused damage to the blood vessels in your hand, and that endothelial lining would be damaged. That needs to be addressed. So those exposed collagen fibers trigger the formation of activated factor 10, and activated factor 10 leads to the formation of prothrombinase, And now we're at the common pathway. Here we can look at the extrinsic and the intrinsic pathways side by side. They often co-occur. They can be triggered by the trauma simultaneously. So let's say you smack your hand into the wall again, and now you've actually torn open the blood vessel wall. Both the extrinsic and the intrinsic pathway will be initiated. The extrinsic pathway happens much more quickly. It requires fewer steps, and it will lead to the formation of prothrombinase before the intrinsic pathway will. Once we have prothrombinase, that leads us to the common pathway. The outcome of the common pathway and the outcome of all blood clotting is the formation of fibrin. The common pathway begins with prothrombinase. And each of these pathways, this superficial overview that I'm presenting really represents a very complex cascade of reactions. So prothrombinase initiates a cascade of reactions that's going to terminate. It will end in what's known as strengthened fibrin threads, and that is the common pathway. Here we see the stages of clot formation. Starting in the upper left, we see an early stage, and we know it's an early stage because we just see a few fibrin threads. They're fairly short, and we can see that they are probably impeding, hampering the movement of some formed elements, red blood cells and platelets. And then moving along the top to the right, we see an intermediate clot because we see more fibrin threads and we see more formed elements that are trapped. And then moving to the bottom left, this is a late stage clot. We see a great density of fibrin there. And on the right, that's a great image. We can clearly see individual red blood cells that have been trapped by this fibrin fibrous protein. All right, so going back to just the bottom portion of that diagram that we were looking at earlier. So whether we arrive at prothrombinase through the extrinsic pathway or the intrinsic pathway, doesn't matter. Once we have prothrombinase, the process will proceed through the common pathway, and the common pathway leads to the formation of an enzyme called thrombin. Thrombin, as you can see from the two arrows extending below the word, thrombin will catalyze two chemical reactions. Looking at the arrow on the left, thrombin will catalyze the conversion of fibrinogen to fibrin. Fibrinogen, there's that suffix, ogen. Fibrinogen is a plasma protein. It's present in our blood all the time. It's produced by our liver. And fibrinogen is inactive. It doesn't have a role. It's activated by thrombin. Thrombin converts fibrinogen into fibrin. Fibrinogen is soluble in water, in blood plasma. Fibrin is not soluble. So it precipitates out of solution and it forms these stringy strands called fibrin. The second reaction that's catalyzed by thrombin, if we follow the arrow to the right, thrombin activates clotting factor 8. And activated clotting factor 8 causes those loose fibrin strands to coil up a little bit. These are proteins, and these proteins can change shape. And as we move from the loose fibrin threads, we get strengthened fibrin threads where that whole clot retracts. It becomes denser. It's more of a mass, blocking the flow of formed elements. So once again, fibrinogen is a soluble plasma protein. It's produced by our liver. It's in our blood all the time, and it becomes activated by an enzyme called thrombin. Thrombin causes fibrinogen to be converted into fibrin, and fibrin is not soluble in water and precipitates out of solution to form this stringy mass. And in this diagram, we can use it to see the overlap, the co-occurrence of these three mechanisms of hemostasis: vascular spasm, the formation of a platelet plug, as well as coagulation, also known as clotting. They all work together to limit blood flow through the wall of a damaged blood vessel. I'll encourage you to stop right now and take a minute and a half to watch this very informative, very cool animation of blood clotting. Hemophilia is a bleeding disorder. It's a genetic disorder, so someone is born with it. And there are many different forms of hemophilia. What they all have in common is the person lacks enough of one or more clotting factors. As I mentioned earlier, there are dozens of different clotting factors. And of course, many genes are needed to code for the production of these dozens of clotting factors. And somebody can be born with a mutation to a gene that leads to an inability or an insufficient ability to produce a clotting factor. And that means that the blood has an impaired ability to clot. Vascular spasm can still happen. A platelet plug can form, although some of the clotting factors also play a role in the formation of a platelet plug, so that might be impaired as well. But a blood clot is unable to form. And so these people have lots of what's known as spontaneous bleeding. They have nosebleeds. They show visible bruising often. And because hemophilia exists, it gives us a real sense of how often our blood vessel walls are damaged, and then clotting happens and they repair themselves unbeknownst to us because we don't have the nosebleeds, we don't have the bruising as we witness in a hemophiliac. So hemophilia can vary in severity. Somebody can have mild hemophilia, or in this case, this individual has severe hemophilia indicated by all of that bruising, and that's not due to any obvious trauma. This person was not in an accident, was not beaten up. They get out of the bed in the morning and this is what they look like. And so we can compare someone who doesn't have hemophilia with someone who does have hemophilia by looking at mechanisms of hemostasis. Up top, as we move from right to left, we can see the process that occurs in someone who doesn't have hemophilia. And down below, we can see the processes that occur in someone who does have hemophilia. So in both individuals, the blood vessel wall is damaged and they start to bleed. In both individuals, vascular spasm occurs normally, and vascular spasm helps to limit blood flow through the damaged vessel. And then when we get to number 3, we start to see differences between the two individuals. Up top, the person who doesn't have hemophilia forms a platelet plug, and they also form a fibrin clot. Down below, the person who does have hemophilia might have an impaired ability to form a platelet plug because there is overlap in signaling molecules in clotting factors. Some clotting factors contribute to the formation of a platelet plug, and platelets release clotting factors. And so this person has an impaired ability to form a platelet plug and, of course, an impaired ability to form a clot. And so notice that this person continues to bleed while the panel up top, for the person who doesn't have hemophilia, they formed a clot and hemostasis has occurred. They are no longer bleeding. So the way that hemophilia is treated is totally dependent upon the type of hemophilia a person has. So for some types of hemophilia, an adequate treatment is to deliver clotting factors into their cardiovascular system periodically. So every few months, the person might go and have a transfusion of the clotting factors that they're missing, and this is sufficient to control symptoms. For other types of hemophilia, we have drugs that are able to stimulate or enhance, promote the formation of clotting factors by liver cells, and these drugs are hopefully adequate to control symptoms. So now that we've talked about how a blood clot forms, we need to talk about anti-clotting mechanisms. And anti-clotting mechanisms have two aspects. Number one, an anti-clotting mechanism is responsible for dissolving existing clots after the clot is no longer necessary, and that's called fibrinolysis. And number two, anti-clotting mechanisms oppose or prevent the formation of inappropriate clots, and those inappropriate clots are sometimes referred to as thromboses. So the fibrinolytic system is in place to get rid of or dissolve clots when they're no longer necessary. So think about the word fibrinolytic. You know what fibrin is. It's that insoluble protein, that stringy mass that forms a that forms a clot. And lysis means to split. So during fibrinolysis, fibrin is chopped up into smaller molecules and the clot is disassembled. And this occurs after the blood vessel wall and neighboring tissues are sufficiently healed. So it starts with the formation of the clot. So immediately following the injury, while the clot is forming, an important molecule known as plasminogen is incorporated into the clot as it forms. And there's that suffix again, ogen. Plasminogen is an inactive precursor to plasmin. Yes, you guessed correctly. And that plasminogen, that inactive precursor, is incorporated into the clot as it forms. So we've seen this image before. This is a blood clot where fibrin is shown in sort of that grayish green color. And here I've added blue dots to help you visualize that that clot includes a different molecule called plasminogen. And while that tissue is healing, that plasminogen is inactive. It's only activated once the normal healthy cells following the healing process release a substance called TPA. So TPA stands for tissue plasminogen activator. I think that's a great name for a molecule. It tells you what it does. It activates plasminogen. And TPA will convert plasminogen into plasmin. And plasmin is active. It's an enzyme, and it will catabolize or chop up fibrin. So this tissue plasminogen activator is produced by healthy endothelial cells after the wall of the vessel is repaired. Those normal, healthy, functioning endothelial cells give this signal, hey, it's okay to break down the clot now. We're all good. The blood vessel wall is intact. There's no risk of bleeding again. In this diagram, we can see the clot. A clot is also known as a thrombus. And our purple molecule is tissue plasminogen activator. And that was secreted by healthy endothelial cells lining that newly healed blood vessel wall. And what TPA does is it interacts with the plasminogen that was built into the clot as that clot originally formed, and it converts that plasminogen into plasmin. And then plasmin is able to degrade fibrin. It can chop it up into smaller molecules, and so it's no longer that mass of stringy mess. And so in this way, the clot dissolves or the clot degrades. The discovery of TPA was monumental for certain types of stroke patients. And TPA can be used as what's known as a clot buster for people who have ischemic strokes. Not all strokes are ischemic strokes. Some strokes are hemorrhagic strokes. And so tissue plasminogen activator is appropriate for one and definitely not appropriate for the other. So in general, a stroke is caused by inadequate blood flow to part of the brain. And what might lead to inadequate blood flow? Here we see the two basic types of stroke. Up top, where we see a blockage that looks like an atherosclerotic plaque. And when that blockage prevents blood flow through vessels that serve a particular area of the brain. We call that an ischemic stroke. Ischemia is lack of blood flow. Down below, we can also deprive brain tissue of adequate blood when we rupture a blood vessel. That's called a hemorrhagic stroke, where to hemorrhage means to bleed. And so because that blood vessel wall is broken open, that blood is leaving the blood vessel and it's not making it downstream to its destination. So downstream of this rupture, some part of the brain is being deprived of oxygen and nutrients, and both of these are called strokes. So let's think about both types of stroke in the same blood vessel. So if the person had a hemorrhagic stroke, if the blood vessel wall broke open and they were bleeding across that vessel wall, then it's labeled here a hemostatic plug. And a hemostatic plug accounts for the fact that it's both a platelet plug and a fibrin clot. And so the wall of that vessel is plugged up to prevent further bleeding. And then we can see evidence of an ischemic stroke. If we move to the right, we see that thrombus. That thrombus is a blood clot. Maybe that clot was an embolism. That's an inappropriate clot. It's existing where no damage to the vessel wall exists, and it's preventing adequate flow of blood through the vessel. So let's consider these two circumstances. On the left, we have evidence of a hemorrhagic stroke. On the right, we have evidence of an ischemic stroke. Which patient would benefit from TPA and which patient would perhaps be harmed by the administration of TPA? Remember, TPA is tissue plasminogen activator, and both of these are clots, and they both include plasminogen. Tissue plasminogen activator will activate that plasminogen and cause the clot to break down or to dissolve. What are the consequences of dissolving each of these clots? What happens if we dissolve the hemostatic plug? We bleed again, and that's a bad thing. What happens if we dissolve the thrombus on the right? We open up blood flow through the vessel again, and that's a good thing. So continuing to talk about TPA for a little while, it's been in use for several decades now, and hospitals have developed evidence-based protocols for determining whether or not TPA is administered to a stroke patient. And primarily, we need to determine what type of stroke the patient has had in order to prevent TPA from causing more harm. All right, so continuing on in anti-clotting systems, another branch of anti-clotting mechanisms concerns the prevention of thrombosis. So thrombosis is the formation of a clot in an undamaged vessel. So there's no need for a clot, and a clot forms nonetheless. And this clot can be dangerous. It can block flow through the vessel, and or a piece of that clot can break off and become an embolus. It can become a piece of debris that's carried around the cardiovascular system. And that debris might become lodged in a smaller vessel and occlude or block flow through that vessel. So that's a dangerous situation. In this patient, we see a stark color difference in their two lower limbs. The limb on the left shows a deep purple bluish color, and the limb on the right, that skin shows a normal pinkish hue. And so that color indicates that this person has suffered an arterial thrombosis in one leg. So a blood clot formed, and it blocks flow of blood downstream of the clot. And so that bluish, purplish coloration is an indicator of ischemia, insufficient blood flow. So what can possibly trigger the formation of a thrombosis, an inappropriate clot? Well, you'll recall that blood is normally only in contact with perfectly intact endothelial lining of the cardiovascular system. So anything that disrupts that intact endothelial lining increases the likelihood that the clotting cascade will be engaged. And so those things include worn out endothelial cells. A bacterial or viral infection in a vessel can actually cause a roughened surface. And atherosclerosis. Atherosclerosis, we'll look at an image in just a moment, but to remind you, is that fatty plaque that accumulates in the wall of a vessel. And then what we haven't talked about before is, should blood become stagnant? Should it stop flowing and start to pool, even in a tiny spot inside one vessel? That slow moving blood has a much greater tendency to form a clot than fast flowing blood. So all of these can lead to the platelet release reaction, and the platelet release reaction increases the likelihood of the clotting cascade occurring. So here we're looking at blood flow through a vessel that has an atherosclerotic plaque. And what's possible for these plaques is that the cap over the plaque can rupture. And so now that blood is no longer flowing across a smooth, undamaged endothelial surface. There's a damaged endothelial surface, and platelets will stick and a clot will form. And here we see that that clot is blocking the flow of blood through that vessel. If that's in the heart, as illustrated in this diagram, it can lead to a myocardial infarction. If this happens in the brain, it will lead to an ischemic stroke. So somebody who is at higher risk for a thrombosis, for the formation of an inappropriate clot, they may be treated with anticoagulant drugs. This is a whole class of drugs. What they all have in common is that they slow down the process by which a clot forms. Perhaps you recognize some of the names of the drugs there: Coumadin, heparin, and aspirin. Those are different molecules, and they all interfere with one or more aspects of the clotting cascade. And so anticoagulants are commonly prescribed to reduce the risk of heart attack and to reduce the risk of stroke, which is also known as a cerebrovascular accident, or a CVA. I highly recommend that you spend the next 2.5 minutes watching this great illustration of how a deep vein thrombosis occurs and how a deep vein thrombosis can lead to a pulmonary embolism, a very common and life-threatening situation. And that's it. We've now wrapped up part 3 of cardiovascular physiology. You just completed segment 2 of part 3, and that concludes our coverage of the cardiovascular system. See you next time.